I would like to welcome our speaker, Chief Medical Officer at Designs for Health and Diagnostic Solutions, Dr. David Brady. Welcome, Dr. Brady.
Everybody. Hey, everybody. How are you? Thanks for joining us this morning. All right, we're going to get going here into the presentation. We're going to talk about some of these issues of the autoimmune epidemic and how that may be influenced by the microbiome. And I guess more precisely, the GI microbiota, a component of the microbiome. As was mentioned in the beginning, my disclosures are just that I am the CMO of Diagnostic Solutions Laboratory and Designs for Health. Okay, well, all of us in healthcare, all of us clinicians, we certainly are witness to the fact that there's an incredible rise of autoimmune diseases out there, particularly in the Western industrialized or developed world. This is not happening in the relatively undeveloped world, and there are some really good hypotheses on why that's the case. But if you just look at epidemiological data, incidence data for autoimmune diseases across the whole spectrum, these include diseases that may have been at one point conventionally classified as Th1 dominant or Th2 dominant autoimmunity. It really doesn't matter. All of these are on such a meteoric rise, as you see in these incidence curves, just for a few of the different autoimmune diseases over the latter half of the 20th century leading into the 21st. This is a meteoric rise. This is the only thing that seems to really move this fast when you talk to epidemiologists. When they look at something like this, they think an infectious outbreak. So we're going to actually look at autoimmunity today framed as an infectious disease, because in many cases, in many instances, not every instance, but in many instances, it is a stealth infectious disorder. And, you know, while today everything is about genomics and genes and all of that, this type of rapid change is not driven by genetics. There are certainly genetics involved in autoimmune disease from the standpoint of predilection toward autoimmunity, various characteristics of the immune response in an individual. There's no denying that. But those genes have to play with the environment. Something's going on exogenous to us, something in the environment that is driving this autoimmune epidemic in concert with our genes. And we're going to look at what some of those factors are. Now, how bad is the problem? It's pretty bad. I mean, when you probably the epicenter is the United States, a Western, industrialized, developed country with a very shaky, adulterated food supply and a lot of environmental issues and so forth. But it's to the point now where if you include asthma and allergies, particularly in pediatric populations, 1 in 2 Americans suffer from an immune dysregulatory disorder. And so that's 50%. And this is probably going to get worse after the COVID-19 experience, because it is pretty much being observed already that post-COVID infection or post-SARS-CoV-2 infection and potentially post-COVID-19 vaccination, we don't know, there may be induction of various types of autoimmune responses. It's likely. Now, how does this touch the microbiota, which is the subject of the whole event today? Well, it's emerged pretty quickly through the whole process of looking and studying at the microbiota, that there are certain prevalences of different types of microbiota and microbiome patterns in different disease populations. So when you look at, a standard population without autoimmune disease and populations with specific autoimmune diseases, and you look into their microbiome, and today we're going to concentrate, like I said, on the GI microbiota, you see differences. Now, are those differences just kind of a coincidence, or are they causal? We're going to look at what the data says on some of that. But if we just back up and look at it just conceptually, we have this hollow tube running through the middle of us, which is technically outside of us, and that's the GI tract. And that hollow tube running through the core of our body is full of some pretty nasty stuff. It's full of a whole host of microbes that outnumber the cells in the body. We all know this. This includes bacterium, viruses, protozoa, helminths, bacteriophages. I mean, you name it, they're there. And it's not only their presence, it's the presence of their metabolites that they secrete, their genes and their genetic expression. And add to that all the food that we put in our mouth every day that goes through that tube as we digest it. There are a lot of metabolites of that. There are a lot of things in that food mass that is not particularly healthy, that could be toxic if it was immediately exposed to our systemic, let's say, blood circulation, lymphatic circulation. So the only thing keeping this slew of material that's potentially toxic to us and even potentially lethal to us, keeping it away from our systemic blood and lymphatic supply and dancing with our immune system proper, is this one cell, layer thick GI mucosa. So we're going to look not only at the composition of what may be in the GI tract, but also what is its ability to actually interface with our immune system, circulatory system, lymphatic system, and how might that drive different immune processes or autoimmune processes. So the composition of the GI tract, what's going on inside the lumen of the GI tract, but also what's going on along the GI mucosa or our protective barrier, are both very important, and we'll touch on both of those things. But just to illustrate how important the composition of the GI microbiota, meaning the microbes that are there, how important that may be to the immune response, I'm not going to show you a bunch of studies and go into real detail on every single study, but I do want to show you this one. This was a very elegant study and a very groundbreaking study. And it was a mouse model study. It wasn't a human study, but they took a wild type mouse, healthy animals, no GI disease, no autoimmune disease. And they introduced into their normal GI microbiota composition one bacteria, and that was Helicobacter hepaticus, H. hepaticus for short. This is related to Helicobacter pylori, same genus, different species. And H. hepaticus is known to strongly activate Th17 cells. Th17 cells are immune cells that produce and release a very, very inflammatory cytokine called IL-17. IL-17 can basically cause an inflammatory storm. And it did so in these animals. And they basically developed a mouse version of inflammatory bowel disease, which is a classic autoimmune disorder. And they demonstrated that. So they took healthy animals, they introduced one bacterium into their gut and produced an inflammatory bowel disease in this animal model. They then introduced one more bacterium, B. fragilis, and B. fragilis expresses on its surface a PSA, or a polysaccharide A. This PSA can be picked up by dendritic cells and presented on their surface, which in turn activates CD4 and T regulatory cells in the animals. And the T regs are sort of the opposite of the Th17 cells. They produce a very anti-inflammatory cytokine or interleukin known as IL-10, which can suppress the IL-17. It's basically the antidote to it, and it alleviated the IBD in these animals. So long story short, they took healthy animals, they introduced one bacterium into their gut, produced an autoimmune disease, which was inflammatory bowel disease or a colitis. They introduced one more bacterium and they basically cured them. So this is very, very interesting study. And is it something that's applicable to the human experience? It turns out that it is. And in fact, many of us in medical school or whatever healthcare training we had, when we were taught about the immune system and various autoimmune diseases, they were often framed in through a more systemic lens, in Th1 dominance or Th2 dominance, which side of the immune system was sort of overrunning the other and losing that homeostasis or balance. But when we look at autoimmune disease and the influence of the GI system and the GI microbiota and the enteric immune response, and when we look at it through a functional medicine lens, we're really looking at it from the standpoint of Th17 and Tregs. So Th17 cells releasing this highly inflammatory IL-17 will push us toward an autoimmunity stance or a hyperimmune response, where T regulatory activity and IL-10 suppresses that and calms things down. So it produces immune tolerance. And we want to find that sweet spot between them. When the scales get imbalanced, that's when we have problems. So trying to do things therapeutically, which we'll talk about today, which try to balance out that Th17 and T regulatory, and finding that sort of a yin yang between those two is of utmost importance. Now, when we look in the medical literature for different systemic diseases and pathologies that have been firmly linked to aberrations in the GI microbiota, or what we may call dysbiosis, and when we look at disorders that are linked toward intestinal hyperpermeability, or in common parlance, leaky gut syndrome, and often both of those things concomitantly present, dysbiosis and leaky gut, because they do go hand in hand the vast majority of the time. There's a lot out there suggesting that when these things are present, there's a much higher predilection of various diseases, most of them inflammatory, many of them autoimmune. So we're going to look at just a few of those connections today with the allotted time that we have. But I don't want to make it sound like these connections are just to autoimmune diseases. That may be the most direct line, but let's look at a very complex multifactorial disorder like autism in children. Autism is a very puzzling disorder, right? And it's very multifactorial. Nobody knows exactly, what's going on with it, what's causing it. We have ideas, we have hypotheses, but there's certainly no universal agreement on causality of autism. But when we use quantitative PCR, so the highest level of being able to determine what is going on in the GI tract in autistic subjects versus normal controls, meaning children without autism or any inflammatory bowel disease or any known autoimmune or systemic condition, we do see differences. The most striking difference, not the only differences, but the most striking differences is the level of Clostridium. And various clusters of Clostridium have been identified as significantly higher in autistic individuals. So what does this mean? Does this mean, Clostridium overgrowth causes autism? No, it's not that simple, because then you can get into chicken or egg. Is the Clostridium clusters elevated because of autism, because of altered eating patterns in autism or other metabolic factors? That's not quite answered yet. Although in various autoimmune disease, we actually have answered what comes first, the change in the microbiota or the disease. And it turns out that it seems like the change in the microbiota precedes at least the clinical expression of the disorder. But if Clostridium and changes in the GI microbiota had significant influence functionally in autistic individuals, then you would think that changing the landscape of the microbiota may alter their actual functional outcomes. And is that the case? Well, there's limited data on it, but probably the most powerful therapy we have to date to change the landscape of the microbiota composition in a subject is fecal microbial transplantation. Like I said, limited studies, but the studies that are there on autistic individuals and using stool transplantation show that the stool transplantation does alter the GI microbiota in a favorable direction, and it does change functional outcomes. It changes behaviors in autistic subjects. So less head banging, less repetitive motions, less self-harm, more social interaction, things like that that are measurable can be observed when we change the landscape of the microbiota. It doesn't mean this is the magical cure for autism, but it certainly is something that we need to continue to research and potentially use to our advantage. But if we get to even much more common things, like just inflammatory disorders in general. So systemic inflammatory disorders have been associated with significant, very predictable changes in the GI microbiota. And this is along with precision immune therapies in cancer. This is probably the hottest area of medical research right now, at least pre-COVID. So when we look at some of this research, you can see that they're starting to develop very precise, very complex, what we call signatures of the microbiota and the GI environment in different disease states. So in this slide, I'm just picking out one. This is for rheumatoid arthritis and just spondyloarthropathies, inflammatory arthritis in general. And what the researchers are mapping is not only which, let's say, bacteria may be elevated or reduced in these disease processes in populations with these diseases versus populations without. It's not only the bacteria or the microbe, whatever it may be, being high or low, it's what results from that. How does this change different metabolites or the metabolome of the GI tract? Does it change, for instance, the relative production of various short chain fatty acids, which have influence over the microbiota in general and the GI mucosal health? How does it change the stance of the enteric immune response? Does it change Th17 or Th regulatory function or Th1, whatever it may be? And does it change cytokine expression? Does it change the kind of cytokines that are out there that can drive inflammation and can drive a disease process? These things are being looked at, quantified, and put out there in the literature for us to be able to use, not only in research to classify different patients, but now to be clinically useful information to help us make therapeutic decisions. And this is research that is not living just in the realm of functional medicine and integrative medicine. This is really permeated into the highest levels of orthodox or standard medicine, including pharmaceutical development. Here on this slide, I just put several of the microbiome drugs that are in development, and these are going at it from different angles, including very specific standardized fecal microbial transplantation material, to designer probiotics, to even beyond the organism, like a designer specific proprietary probiotic strain, actually something more reduced, because pharma likes this, ultimate reduction of what is the actual therapeutic active agent. So if you look on this slide, I know it's probably a little small for you, but the third up from the bottom is symbiotics. This is actually using that literature I showed you in the beginning of the presentation on introducing B. fragilis into those mouse models, and the PSA that it expresses being taken up by the immune system and basically shutting off the inflammatory bowel disease. This is actually being developed in a drug model. So the PSA from B. fragilis is being developed as a drug, and it targeted for inflammatory bowel disease and even for multiple sclerosis. So pharma is on this big time. And what this means for some of our therapeutic interventions, such as probiotics, prebiotics, from a regulatory standpoint, once these things start being developed through drug pathways, will be interesting to watch. But in order to actually apply this stuff, we have to know what's there, right? What is in the GI tract in these individuals with these different diseases? What is there in a patient that we're dealing with in our office? And all of this research has been made possible by an evolution in our ability to diagnostically or to in an investigational way find out what's going on in the GI tract through stool analysis. Now, when I started in this, I've been in practice for 30 years. We didn't have molecular type of technologies at our disposal, particularly as clinicians, and not even in research at that time, to look at stool material and try to find out what's in the GI tract. It was all microbiology, right? It was all plating things out, growing it, trying to typify it, trying to classify it, which had severe limitations. In fact, you couldn't really do that in any tangible way with anaerobic organisms, which make up the vast bulk of what's in the GI tract. But the molecular revolution, PCR, things like that really changed the game, changed the landscape. And in a recent article in the Townsend letter, in fact, an edition that I was actually featured on the cover for my work in this realm over the years, I put together an article with Cass Nelson Dooley on the different methodologies in stool testing and what are the differences and when is one better than the other. And it covers all the different ones out there, from quantitative or real-time PCR sitting at the top to all the way down to microscopy and everything in between, shotgun metagenomic sequencing, next-gen 16S sequencing, and so forth. They all have a place. They all have a strength, they all have a weakness. And we're going to try to talk about that a little bit today within the context of the time that we have. However, that article is available It's available on my website in the articles tab at drdavidbrady.com, just drdavidbrady.com. But it is also available on the Townsend Letter site. So if you just go to the, just Google Townsend Letter, go to their site, you can find this article. It was in the January of this year's edition. But basically, when we're talking about testing and stool testing, using molecular or DNA, I'm not even really going to talk much about microbiology because that's really old school, got a lot of limitations. I think everybody understands that by now. But I think clinicians are very confused at the different types of molecular or DNA-based ways to look at stool. The two major ways are quantitative PCR or next-gen sequencing. They both have value, they both have their place, but they're very different, and they're used for very different reasons if used properly. And examples, quantitative PCR, actually looking at specific organisms, getting an actual specific, very accurate amount of them, that's where the quantitation comes in, is used diagnostically. This is like something you see on the GI map test from DSL. On the other side of the equation, we have more broad, wide lens sequencing, like 16S next generation sequencing. This is what Ubiome, the very popular direct-to-consumer stool test that was out there for a while. Now it's off the market, but there's others. Certainly most of what's out there uses 16X next-gen sequencing. We'll talk about the differences there. And basically, just so you can frame it up, I made this slide. Next-gen sequencing is very good if you want to get way high up, like 30,000 feet, and look down at everything that's there. So if you want to see the topography for a wide, area, like is shown in this slide with a picture out of the jet window, that's next-gen sequencing. Next-gen sequencing or shotgun sequencing, shotgun, right? Big spray. We're looking at everything that's there, not specifically quantifying it. So we're not getting a lot of clarity. We're not getting a lot of resolution, but we're covering a large ground. So it's like looking down from way, way above. If you want to see, hey, what is the general gist? Like, is it mountains? Is it plains? Is it valleys? Is it rivers? Then next-gen sequencing is great. And if you're trying to answer research questions like, what is different about the entire composition of the GI microbiota in rheumatoid arthritis patients versus patients without, or patients with diabetes versus patients without, or even patients in Tokyo, or people in Tokyo versus people in Los Angeles. Then you use next-gen sequencing, you get reams of data about organisms that you've never heard of before, and we don't know have any clinical significance or not, but you're mining data. This is a big data play, and it's used in big data research. You try to take lessons from that clinically and bring it to the individual. But when you're testing on the individual level, you then want to look for those specific targets that you were taught to look for from the big data sequencing research. But as a clinician, trying to make a clinical decision on a patient, such as, is there a pathogen there and do I need to use an antibiotic or a natural antibiotic, or do I need to take an action? You really want to get down at a lower elevation. You may not be looking at as much ground or as many organisms, but you're looking at a curated, targeted list of organisms, but getting very specific information on that quantitative data. How much DNA, how much organism do I need to treat? Is it crossing what we would consider a clinical threshold where it's something to worry about? Those are the differences. From the macro perspective, there are many, differences technologically. But if you look at a next-gen sequencing test or a 16S classic test, which I'm sure pretty much everyone listening has, you will usually not know what to do with it, right? It's just page after page after page of organisms after organisms that you never learned about. You don't know what they are. And you know what? Nobody knows what most of them do. They're not, most of them, we have no idea if they're clinically significant or not. And we're assuming they're not overtly clinically significant, or we would know about them by now. Do they cause profuse diarrhea? Do they cause gastroenteritis? Do they cause, whatever it may be? Now, these reports are often given to you, and they're reported as a percentage of the total DNA recovered. How much of that organism's DNA by percentage did it make up of the entire DNA catch? That's not quantitative, that's relative data. So it's all looking at organisms in relative amounts to one another. It's not quantitative. Where when you look at a quantitative PCR test like the GI map, you're getting very specific ranges and you're getting quantitation. You're not getting nearly the number of organisms because the test by intention is looking at a very curated list of organisms that we know have clinical significance, either overtly from the standpoint of this first page here shown are pathogens. These are organisms, first bacterial, then parasitic, in this case protozoal, and viral, that we know can cause acute diarrhea, gastroenteritis, food poisoning, and the like. In this specific example, for instance, you see that this patient had Clostridia difficile DNA, but not just DNA to identify the organism C. difficile from the genus and species standpoint, but with quantitation, we can actually look much deeper at functional characteristics of the genomics. So here we see identification at a significant enough level of the genes in the C. difficile population that are capable of producing the toxins associated with the diarrhea and the gastric distress and the inflammation caused by C. diff. This is toxin A and toxin B. Now, in many cases, we see this at low levels in patients who are asymptomatic. They don't have acute diarrhea. They don't have what appears to be an overt Clostridia difficile infection like a post-antibiotic diarrhea, for instance. But we know that these patients harbor a small amount of C. diff as a population, and that population has the toxin A and toxin B producing genes, and they can be switched on. So this would be the type of patient that would be much more likely, for instance, to have diarrhea if they're given antibiotics, and much more likely to go on to potentially develop pseudomembranous colitis with antibiotic therapy. But we can use this same quantitative technology beyond just pathogens because, you know, hospital pathology labs and so forth, they can use this technology now and they will tend to test pathogens in patients with acute diarrhea and so forth. But they don't do what we need as a functional medicine physician or clinician, which means we don't only want to look at overt pathogens. We want to look at conditional pathogens, commensals, opportunistic organisms, and so forth. And that's what you get on something like a GI map. You can see here in this same patient, they had an elevated H. pylori, population identified above clinical threshold. And beyond just having higher levels of H. pylori, we can look into the genomics of the H. pylori population, and we find out that the genes are present to produce 4 of the most significant virulence factors. The virulence factors, are the metabolites of these organisms, are what actually drive the inflammation and the erosion of the gastric and the duodenal mucosa and elevate the chance for gastric cancer, for instance. So we can determine, is it a low-level benign H. pylori that really does not need clinical action, or is it something significant to treat? Further down, we see commensals, or normal or beneficial organisms, if you will. And what you often see when there's pathogens present or opportunistic overgrowth is a corresponding decrease in some of the commensal organisms, including some of the keystone species here, like Fecalobacterium prusnitzii, changes the whole metabolome in the gut, lower levels of short chain fatty acids and butyrates and things such as that. So we can look at commensals, but we can look at opportunists. And I picked this sample specifically. It's actually a sample on a patient, because this patient had a little bit of toxic genes with those C. difficile. They had H. pylori, low commensals, but elevated opportunistic organisms. Opportunistic organisms aren't overt pathogens. They're not the good guys or the commensals. They're everything in between, which makes up the bulk of the microbiota. And if they are given the opportunity to overgrow for some reason, maybe low digestive function, low digestive enzymes, changes in pH, whatever it may be, they can grow to a higher level. We get a higher DNA catch. They can actually stimulate problems. It could be functional, what is often considered functional bowel problems and diagnosis as IBS, constipation dominant, diarrhea dominant, so forth, or they can be associated with the genesis of autoimmunity. And in this sample here, in this second little bucket here, potential autoimmune triggers, we see organisms like Fusobacterium and Klebsiella, which we're going to talk about specifically as it relates to autoimmunity. We have candida and different fungal organisms. We even have viral organisms like Epstein-Barr virus and cytomegalovirus, which can be identified in the stool. And when they're in the GI tract, they're also associated with higher levels of autoimmune activity, just like when they are found in the systemic with antibody testing in blood. We can look through a more comprehensive list of parasites, such as the smaller ones, protozoal organisms, plus larger organisms like helminths, and then actually do stool chemistries, which you don't get stool chemistries on sequencing tests because they're not meant to be diagnostic. They're not meant to be used in medical decision-making per se. So in this case, this person had low elastase 1. They had low pancreatic excrement output. They had low enzymatic function. They had a digestive deficiency, dysbiosis. And in fact, persistent infection with H. pylori is associated with low pancreatic enzyme output and hypochlorhydria, which can lead to opportunistic overgrowth. So it's a classic pattern we see on GI map. You then will get changes in the metabolome in the gut, such as overproduction of beta-glucuronidase, which can deconjugate toxins being shed through glucuronidation, such as estrogen, which is important in females, especially those with genetic breast cancer risks. But here, this person had an elevated anti-gliadin IgA, so they're probably eating gluten and they're not immune tolerant to it. They have a bit of inflammation, as shown by a mild to moderate increase in calprotectin, and an elevation in zonulin, which can contribute to hyperpermeability or leaky gut. So armed with this type of diagnostic information, and in this case with quantitative PCR, we can actually look into the genomics of the microbiota and determine, and particularly in this case, the H. pylori, and determine if you chose to use antibiotic therapy to treat, let's say, this H. pylori infection. You didn't go with natural agents. You wanted to use antibiotics. This guides you into picking the ones that the resident population does not have antibiotic resistance to. So you're not fostering an even more super population, you're actually eradicating it. But back to this latch between the GI microbes and systemic pathology and autoimmunity in particular, it's all over the literature. So I'm giving some of the examples here. Most of the things on this list, if not all of them, I think on this list are autoimmune. We're going to, just for the sake of time we have today, we're going to concentrate on the ones toward the top. Things like autoimmune inflammatory arthritis, like rheumatoid arthritis, ankylosing spondylitis, and the like. And we're going to look at organisms such as Klebsiella, Citrobacter, Proteus, Prevotella, because it will be a lesson that can then be extrapolated over other associations in autoimmunity. And what happens when a bug overgrows, and how can that potentially cause an autoimmune response? There are multiple answers to that. You can have metabolites of the bug or genetic expression of the bug altering enzymatic function, modulating receptor function, and so forth. But probably the most well understood and the longest understood is a phenomenon called molecular mimicry. And it's a very simple concept, very technical in the specifics, very simple in the concept. And that is if an exogenous peptide or protein is introduced into the body, this could be in a food, could be a food protein. Therefore, certain food proteins in certain individuals can foster autoimmune. We see that certainly in gluten and celiac patients, right? That's an overt example. There are a lot of subtleties of that. But the other thing that presents foreign peptides and proteins to the body is all of the microbes that make up the microbiome, right? The bugs in us, around us. But there's nowhere that there's such a concentration of a diverse level of microbes that we're exposed to than our GI tract and our GI microbiota. These bugs express on their surface proteins and peptides. And in fact, our body's immune system uses that to understand them, identify them, and target them if necessary. But if one of those proteins or peptides is very similar in structure, or the amino acid motif or sequence to a protein or a peptide that's naturally on one of our tissues, there's a potential there for sort of misidentification, for friendly fire. If a microbe overgrowing in our gut stimulates our immune system to key on a certain peptide to identify that microbe and take it out, but that microbe has a protein on it that's very similar in structure to somewhere in the body, in a tissue, the immune system, particularly if it's in a overaggressive state, the patient is in an inflammatory state, they have certain aspects of their immune response, like an HLA pattern, that will make them more efficiently present that antigen and cause a ruckus about it. You can get a cross-reactivity and a friendly fire to a host tissue. So that's the whole concept of molecular mimicry. And this is where genetics and the environment line up, because usually the person has to have the genetic predilection. Like, let's say we're talking about rheumatoid arthritis, inflammatory arthritis, such as an HLA pattern like an HLA-B27. In that case, if they have an overgrowth of Klebsiella or Citrobacter or one of these organisms that produces these peptides, that the HLA-B27 characteristic will cause it to present it out to naive T cells and say, hey, learn this peptide structure, and when you see it, we need to develop an aggressive response against it. Just so happens that those peptide sequences are very similar to peptides or proteins expressed on the synovium of the joint structures. So you can get a misidentified response against joint structures. And one of the best explanations of this is back to an older paper, actually, from well over a decade ago in mucosal immunity in this paper here. And it talks about overgrowth of bacteria, expression of peptides on their surface, concomitant hyperpermeability, HLA-B27 pattern, cross-reactivity, and through molecular mimicry, are an erosive inflammatory destructive response against the synovium, resulting clinically in what we would call rheumatoid arthritis. So on GI maps, we see these patterns all the time. This is a patient who came in with newly diagnosed rheumatoid arthritis from an academic center of excellence, a rather young patient. They wanted to put them on methotrexate and potentially other biologic agents, TNF-alphas and so forth. The patient read on the internet about those immune modifiers, such as opportunistic infections, lymphoma, leukemia, and so forth. They wanted to go a different route. They found functional medicine online. They found a lecture by me or something, and they're sitting in my office. When we GI mapped them, we found overgrowth in the GI microbiota of Citrobacter, Klebsiella, Proteus. So these are some leverage points that we now have to try to clean up the triggers to their immune response, to not necessarily cure their disorder, but to slow it down, to take the oxygen from the fire. Now, over the years, as we've worked with GI map testing, we've been following the literature very closely. So when information came out on Prevotella being very strongly linked to new onset rheumatoid arthritis diagnosed patients, these are being added sequentially to the autoimmune triggers section on the test. So here is a patient subsequently down the road with Prevotella cobri being identified quantitatively higher than the average person. Is this the sole factor driving their rheumatoid arthritis? We don't know. Probably unlikely, but it's one of them. And what we're trying to do is take the straw off the camel's back, right? They also had Proteus mirabilis, which we'll talk about in the beginning. But I did want to back up and remind everyone that the GI tract starts in the mouth. And poor dentition, poor gum health is common. One of the first things we do when a patient comes in the office, in any physical exam, but particularly those with autoimmune disease, and particularly those with inflammatory joint autoimmune diseases, is I do a good oral exam. I look in their mouth. And if they have any kind of gum disease or gingivitis, we get that out to a dentist, to dental hygienists, get it treated. The most common bug to cause periodontal disease, at least in the Western world, is P. gingivalis. We know that P. gingivalis actually produces an enzyme called PAD4, and PAD4 can cause citrullination of host peptides like collagen, vimentin, fibrinogen, all things that are located in the joint, right? So could this organism, by virtue of secreting an enzyme, modify host proteins in certain structures, which then produces a protein on a host tissue that is not recognized any longer as us. It's called a hapten. That happens all the time. So this is not molecular mimicry. This is a bacteria producing an enzyme which will citrullinate host peptides. We see this with P. gingivalis. And when you learned laboratory diagnosis in medical school and you do a rheumatoid panel, one of the things you're looking for, besides things like rheumatoid factor and C-reactive protein, SED rates, things like that, is CCP, right? Cyclic citrulinated peptide. You're actually looking for the end effects of this citrullination being driven by bacteria. But most doctors, when they order this lab test, when they see it high, they're taught, oh, if this is high, think rheumatoid arthritis, but they don't really understand why. So this is why. This is how the citrullination happens. And it's not only something with joints and RA. We see citrulinated peptides, probably from bacterial expression of enzymes that modify host proteins in Alzheimer's disease, in brain lesions, in atherosclerotic lesions. So how much of this effect is driving other serious chronic diseases is just now starting to be known. So we look in the mouth too. We do oral swabs and do PCR on that. And in this case, this person had high-risk pathogens identified overgrowing in the mouth, P. gingivalis, but they also had a couple of moderate-risk pathogens, including Fusobacteria and Prevotella. This was a rheumatoid arthritis patient. So this is our opportunity in functional medicine to clean up the patient's environment and milieu to make them less likely to drive their disease process forward. And this really, all of these concepts started with very early researchers, and this one of the seminal researchers is Alan Emberger from England, and Sir Emberger looked at rheumatoid arthritis as an infectious disease decades ago. In fact, you know, before we had the biologics and the TNF alphas and all these immune response modifying drugs, the generation of drugs used in rheumatoid arthritis before that were called DMARDs, disease-modifying anti-rheumatic drugs, and they were by and large antibiotics. They were first used in an off-label manner because when it was observed, just clinically, that when patients with rheumatoid arthritis were given certain classes of antibiotics for other reasons, they had an infection of some sort, so they were given an antibiotic, came in and told the doctors, hey, my RA is better. I haven't felt joint pain since you put me on that antibiotic. So doctors started using it, sort of off-label, then it got approved for it, and we had the whole class of DMARDs. People like Emberger were like, hey, there's something to that. Is it the antibiotic properties of these drugs that are working? And he's put out a multitude, I mean, just more papers than you can count on this type of phenomena. He started saying, listen, if the antibiotics are working, is there an infection somewhere that we're just not noticing or seeing? Is it a stealth subclinical infection? He remembered that there's a strong female predominance in RA. So he started asking himself, what kind of infections are females much more prone to than males? He said, urinary tract infections. He started looking in the urine and going beyond just standard commercial testing of urine for infection and culture, actually developing much more sensitive research assays. And he identified an organism that was prevalent in the women with RA and almost never found in the women without. And that was Proteus, in fact, Proteus mirabilis. And he kept following down that tunnel, and he found out that the bacterium Proteus mirabilis produces a bacterial hemolysin and urease enzymes that have a molecular structure that can cause crossover molecular mimicry to specific joint structures, which is pretty amazing. And he worked out all of the molecular, spatial, you know, all of the moieties and all of the motifs, the amino acid motifs, I should say. And he answered many of the questions that we learned in medical school about RA. Female predominance. Well, females have a lot more urinary tract infections. They're more susceptible to it anatomically. There's a higher incidence of RA being diagnosed in women in middle age or just after pregnancy. This is right after there's potential more anatomical disruption to the urinary tract and the urethra, making you more potentially susceptible to an infection. The rheumatoid factors and the other kind of immune response diagnostics we're using are usually secondary phenomena to an immune response, to a B cell stimulation against antigen antibody complexes created from an infection. But the real aha moment was when he actually worked out the amino acid sequences between the Proteus hemolysin and the Proteus urease. And the Proteus hemolysin had an amino acid motif almost exactly matching the HLA pattern in these patients, the HLA-DR molecules unique to these patients' immune response. But the Proteus urease matched a specific type of collagen known as type 11 collagen. Type 11 collagen is only found in certain places in the body. And wouldn't you know, it's found in the places in the body that RA affects, like small joints of the hands. It's not in the larger joints spared by RA. So didn't everybody wonder back in pathology class, why does RA only affect these certain joints and leave the other ones alone? Well, Emberger kind of was onto it. And we see this pattern a lot. We look for Proteus mirabilis in the gut because how do you think it gets into the urinary tract in females? Through the gut. Fecal vaginal contamination generally. So we look for it in the gut. And then studies have shown you can look for it in the gut. You don't have to look for it necessarily in the urine. So how did Emberger treat these patients? He didn't jump and still, his people who follow his work still today, they don't jump to methotrexate, TNF, anti-TNF alphas and things like that. They do what you would think is more like naturopathic medicine, right? They'll use antibiotics. They tend to use antibiotics I avoid, like Cipro and the other the chlorofluoroquinines and all of those ones that are very, very toxic kind of antibiotics we tend to avoid. But he says herbal diet, cranberry juice, high dose vitamin C, right? Acidification of the urine. And then he'll use DMARDs. He'll use antibiotic, penicillamines and chloroquines and things like that. Minocycline, very, very common DMARD, which is an antibiotic. I still have patients today that the best response they get in their severe RA is with minocycline, and they don't get that same kind of clinical benefit with all of the modern immune modifying drugs. Those are the rage. Those are what are being prescribed. They're the newest toy. They're on active IP. They generate the most, money for the pharmaceutical industry. So everyone's off DMARDs onto the new stuff, even though in many cases the old stuff may work better, may be less toxic, and so forth. But that's a whole nother story. So that's just interesting, more connections. Thyroid connections, we've known for a long time, molecular mimicry has been implicated in autoimmunity against the thyroid. Many different organisms, but we know, for instance, Yersinia enterocolitica has on its surface peptides that look a lot like TSH receptors. You can get cross-reactivity between the two, and through molecular mimicry, the people with overgrowth in Yersinia and the right aggressive HLA pattern, they can actually start attacking their own thyroid and can end up being diagnosed with Hashimoto's and or Graves, some form of autoimmune thyroiditis. In fact, this is a patient, fairly young patient, came in actually she came in for weight loss. She was all of a sudden gaining weight, didn't know why. On history, she was fatigued, she was cold all the time, her hair was getting thinner, all the classic thyroid. She was constipated at IBS. She had enterocolitica. When we did her thyroid panel, her free and total T4 were overtly low. Her free and total T3 were very, very functionally low. They were just barely in the normal range. And she had elevated thyroglobulin and TPO antibodies. This was a patient that went to her, standard PCP and everything else. They didn't pick up that she had a thyroid condition. It was pretty evident. And we treated it as such. We tried to clean up the gut. We tried to clean up the leaky gut, the microbiota, tried to clean up her diet, do all those things while we actually had to intervene and treat her thyroid with a combined T4, T3 HRT. So what do we do when we find these sort of aberrations in the GI environment, in the microbiota, overgrowth of opportunists, or even pathogens that we think we might be able to treat in a natural way without resorting to an antibiotic? regardless really of the organism type, bacterial, fungal, protozoal, we tend to use natural antimicrobial options, and we use them very smartly in the right combination to cover all of the sensitivity spectrum that's required to get the job done. This is an actual formulary antimicrobial I designed years ago. I've modified it and tweaked it many times throughout the years based on a lot of the data I see on stool analysis. And this has a lot of your standardized, very typical, long known effective GI antimicrobial botanicals, things with a lot of berberine in them, like barberry, berberine sulfate for Berberis aquifolius, artemisia from standardized artemisia or Chinese wormwood, grapefruit or citrus seed extract, caprylic acid from magnesium caprylate, Juglans niger or black walnut. But there's a novelty in this, the Tribulus terrestris. Most people know that trying to get more testosterone production doesn't work very well at doing that, but it's a great GI antimicrobial. This is actually GI Microbex from DFH. Works very, very well, particularly in combination with a good oil of oregano, like the organic oil of oregano I use from DFH, small little capsules or small soft gels with the herbal formulary one in capsules. You have to dose this the right way. Usually in an adult, it's usually 2 capsules of the multi-herbal, 2 capsules of the oil of oregano, taken away from food, usually at least twice, but usually 3 times a day over the course of several weeks, 3 to 4 weeks. Meanwhile, we're giving a good probiotic with meals twice a day, trying to keep the good guys coming in. And that can usually nudge the GI microbiota in the right direction without wiping everything out. So it's not like dropping, an antibiotic there. Even a gut selective antibiotic like Xifaxan, which is used a lot in SIBO, tends to temporarily make people feel better, and a month later they're back where they started because it just blows everything up and you can't reconstitute a good microbiota. The botanicals, the volatile oils, are interesting in that they tend to take out the pathogens and the opportunist at lower dosing than it takes to get the commensals. So they're perfect for this application. This is an example of someone on the other side of the fence. They have low commensals. Now they may have dysbiosis that needs to be treated as well, but with low commensals, then you're going to get inflammatory cytokines running wild. And this is where we'll often use, in parallel to GI testing or stool testing, a serum-based cytokine panel. So this is CytoDX from DSL. We can look at the pro-inflammatory and anti-inflammatory cytokines. Now, when we developed this panel, we were doing it toward, trying to manage autoimmune patients and stuff from a functional medicine. I've probably done more of these in the last year, trying to early identify cytokine storms and things like that in COVID positive patients. But probiotics are great inflammatory modulators and calm things down. So we tend to use very specific, high dose. This is 100 billion CFU, but it's 10 strains, strain specific, research on every strain. It's not your average ordinary probiotic. This is a ProBiomed 100 from DFH. Sometimes we'll use symbiotics, which blend the probiotics with prebiotics, like xylo-oligosaccharide is my favorite one. Doesn't cause as much gas and bloating. And also Saccharomyces cerevisiae boulardii, particularly like in the example I gave with the C. diff, because the Sac B will actually, the organism will actually gobble up and eat the toxin A and B metabolites produced by C. diff. And then getting people back to probiotic laden and prebiotic laden foods in their diet is the long-term strategy. And the last thing I wanted to talk about is the intestinal barrier. We all know of Professor Fasano's work with zonulin and his work in gluten patients, but we have a lot of markers on the GI map test trying to look for leaky gut. Things, direct markers such as zonulin, which is high in this case, calprotectin for inflammation, but other clues such as anti-gliadin secretory IgA. People are eating gluten that are not fully immunocompatible with it will tend to develop an anti-gliadin IgA elevation. This is an example of something that can kind of eat away at the gut lining over time. And zonulin can be overproduced in people just genetically. These tend to be people with early onset autoimmune disorders like juvenile onset diabetes and so forth. But most zonulin overproducers, the zonulin overproduction is being driven by dysbiosis, by gut bugs. We know certain bugs, particularly overgrowth of E. coli, salmonella, and other common opportunists, can drive production of zonulin. Low levels of bifidobacteria and commensals can also drive zonulin. And we know giving bifidobacteria, for instance, can lower zonulin, but lower even the reaction of celiac patients to gluten. So this is a example of a GI mucosal repair formulation. A lot of mucilaginous botanicals, things like slippery elm, aloe, marshmallow, chamomile, okra, cat's claw. These are all slimy herbs, mucilaginous herbs. And they used to be used because it was thought that the sliminess, the mucilaginous component, kind of coats the gut and lets it heal. A little bit of that may be true, but really you're using glycobiology. It's the mucopolysaccharides in these botanicals that can block antigen antibody docking and actually block immune proliferation. So in addition to L-glutamine and things that feed the gut enterocytes, you get a lot of these immune blocking agents, including, besides the botanical ones, N-acetylglucosamine. Now, N-acetylglucosamine is a very interesting agent. It's being used in MS and other autoimmune disorders, works on a different mode of action than other drugs. It's available in the supplement channel, but it basically is a mucopolysaccharide that gets in the gears between the antigen, between the activated T cell and the antigen, so they can't dock. So this is it graphically. Here you have an antigen-presenting cell presenting the antigen to an activated T cell. When they dock, you get immune proliferation that drives autoimmunity. Taking these mucilaginous botanicals in the gut and the N-acetylglucosamine sort of gets a lattice of these mucopolysaccharides around these docking sites. So the antigen-presenting cell with the antigen just bounces off. You don't get immune proliferation. You slow down the immune response. So we use those therapeutically a lot, like in powders like GI Revive. It's also in capsules, but it's a great product designed along this research. And I always like to end with good news. There's some great research out there that really high quality, good coffee has great influence on the immune system. Drinking coffee all day long, particularly crappy coffee from Dunkin' Donuts or something, will erode your GI lining, cause gastritis, not good. Selectively, really good quality coffee and a really nice piece of organic dark chocolate, both are good for your immune response. So that's your little end with good news. To this, you can really take a deeper dive into a lot of these concepts than I've been able to get to today with many of my papers, including Open Journal of Rheumatology and Autoimmune Disease, some more stuff in the Townsend and other articles. These are all available on my website, once again, drdavidbrady.com. They're all free access as well. You can find them on the actual publications or the journal's websites also. So that's it, and I'll take a few questions with whatever time I have left.
Excellent. Thank you, Dr. Brady, for a great presentation today. It's now time to move into our Q&A session, and I believe we have time for a question here. What is the main natural GI antimicrobial product you use to treat dysbiosis and or SIBO?
Actually, it's the formula I showed there. It's two in combination. I tend to use something called GI Microbex, which is dry powder in a capsule, many botanicals standardized, along with an organic oil of oregano. You can't mix the two. One's oil, one's powder, one's dry, one's wet. But in combination, they'll hit that whole spectrum you want to get to deal with most situations of dysbiosis, including upper GI dysbiosis, which we call SIBO.
Excellent, thank you. And that is just about all the time that we have for today's program. And thank you again to Dr. Brady for a great presentation today. This session was brought to you by integrativepractitioner.com and sponsored by Diagnostic Solutions.